All right, welcome back, everyone. So this is going to be part two in our muscle lecture. And so we left off talking about myofilaments and myofibrils and the hierarchy and the organization of our skeletal muscle. So we said that we spend most of our time at the level of the myofilament and the myofibril, right? So what these myofilaments are proteins, right? So if you recall, proteins are made up of amino acids. We talked about them when we did our cell lecture. We did transcription and translation. So if that's new to you, go back and review that. So these proteins, these are our actual contractile proteins, or at least two of them are contractile proteins, all right? These are what are actually moving past each other, they're sliding past each other, that result in the large scale gross contraction that we see at a very large scale, okay? At a very superficial view. Whenever you look at something and you see a muscle contracting, it's really happening down at this level of the myofilaments. So in general, we have two types. So we have two categories of myofilaments. One is called thick and one is called thin, all right? So first off, the thick filament. The thick filament is referred to as myosin. Myosin, to me, it resembles a golf club. And so if you look at this up here, this is one individual myosin molecule. You have a tail, which you could think of as like the shaft of the golf club. You then have a head, which is like the head of the golf club. For one large thick filament, you add a bunch of these and you combine a bunch of these individual myosin molecules together, and you get this one large thick filament. Okay. Now, what's particularly important about this myosin molecule is the head. The head contains a region, it contains a site where ATP is able to bind, and for reasons that we'll get to later and we'll discuss later, you have to have ATP bind to this myosin head in order to get skeletal muscle contraction. Right, so it's extremely important. The head is what's going to pivot and slide the thin filament over itself, and this causes the muscle to contract. All right. So the second filament, or the second type or category, is the thin filament. All right. The thin filament has a few more details that we'll talk about. So the primary filament that we'll talk about that's making up the thin filament is actin. Right. Actin filament, actin protein. There are two, I guess, types of actin, if you will. There's what's called G-actin and F-actin. All right. The one that we'll kind of refer to here is the G-actin. So the G-actin, the G stands for globular. Okay, globular. So each of these, if you look at this kind of top image, these red balls here, these are individual G-actin proteins. And all these G-actin proteins form a bond and they bind together and they form what's called F-actin. F stands for fibrous, right? So you get one large fiber, if you will. So the G-actin, the individual G-actin proteins or molecules have a site that is being covered by two other proteins called troponin and tropomyosin, right? So again, if you look at this top image, this odd-shaped blue protein is called troponin, and this green one that's wrapping around is called tropomyosin, right? You can see the same thing down here. This purple one is troponin. This green protein is tropomyosin. The troponin and tropomyosin are referred to as regulatory myofilaments, okay, not contractile myofilaments. The myosin and the actin are referred to as contractile myofilaments, all right? So the reason these troponin and tropomyosin are considered regulatory is because they regulate the activity of actin and of myosin, all right? And so what you have is on these actin molecules, right? So what we said is these G-actin, you have a small little site that allows the myosin head to bind. So we said earlier the myosin head binds the actin. If you look on this top image, you should see these little kind of dark regions, these binding sites for the myosin head. Ordinarily, whenever the muscle is relaxed, those sites are covered by this troponin tropomyosin complex, right? So this is where we get troponin tropomyosin regulating the activity of actin and myosin, right? For reasons we'll talk about later, whenever calcium binds, we'll see that this complex moves, exposes the site, and allows the head to then bind, and then you're able to then, at some point, get an actual contraction and get these proteins moving. Okay, so you can take all of these proteins, all these actin and myosin and whatnot, and you can organize them into something that looks like this. And so this is called a sarcomere. All right, so this image that we see right here on the left and the image that you see over here on the right, these are the sarcomeres. The sarcomere is the contractile unit, the functional unit of skeletal muscle. So what's actually moving and contracting whenever we are and producing tension, whenever we contract a muscle, is the sarcomere itself, right? So you define a sarcomere by its boundaries, and these are called Z lines, or sometimes referred to as Z discs. So you see a Z line here on this image to the left, you see another Z line here, you see a Z line here on this image, this histology image, you see another one right here. Everything in between from one Z line to another Z line, that constitutes one sarcomere, all right? So those are the boundaries, all right? You can then look at the sarcomere a bit closer, or a bit more closely, and you can see that there are different regions of the sarcomere that we'll call bands and zones. So there's some that are called zones and some that are called bands. So if we look at these, the first two are called an I and an A band, all right? And I'll give you a helpful mnemonic to help you remember these in just a second. So the I band is only going to have thin filaments, right? So that begs the question, what is a thin filament? The thin filament is the actin, right? Along with its smaller regulatory proteins, troponin and tropomyosin, okay? The A band is thick and thin filaments, right? So we have A band right here. So we have these thick yellow filaments along with these smaller, more thin red filaments. So you get both. You then move on to something called the H zone, and this is only thick filaments, so only myosin, okay? So only these thick yellow filaments that you see here in the middle. And then the M line, this is where the thick filaments attach in the middle. So here's one Z disc, here's one Z disc, so this is one sarcomere, and right in the middle, you have your M line, okay? Now, the mnemonic that I told you I'd give you to help you remember these is relatively straightforward and kind of easy to remember, and I like it. I didn't come up with it, but I really like it. So it helps you to remember which zones have thick, or bands have thick, which ones have thin, and which ones have both. All right. So if we start with both the I band and the H zone, right? If you think of the letter I in comparison to the letter H, the letter I is more thin. It is a thinner letter, so it only contains thin filaments, okay? The H zone is a thicker letter, it's more wide, and so it only contains the thick filaments. And then the A band, you can think of as an intermediate. It's somewhere between I and H, so it contains both thick and thin filaments, okay? All right, so that's a little mnemonic that I like to use, okay? So the whole reason we're talking about these different bands and zones is that whenever we contract, these can change size. And so, you see, whenever we contract and we pull these together, our Z discs are going to get closer together, so the sarcomere is going to shorten. We can also pull our actin in, and we can take away our H zone. And so we'd have, instead of just thick filaments, you could have both thick and thin filaments. And so you can look at a sarcomere on like a schematic or an actual image, and you can see, okay, is this muscle contracted? Is it relaxed? Or to what extent is it contracted or relaxed? All right. So we're moving forward to talking about how we actually get this sarcomere to actually contract. Right. So how do you get the sarcomere to actually shorten? And the way you do it very rudimentarily here is you take your neuron, you take your motor unit that we'll talk about in a second. You take this neuron and you send an impulse down the axon, and you send it to a grouping of muscle fibers. All right. So you have a motor unit in the brain. So as we'll talk about later, we have a region called the precentral gyrus of the frontal lobe that is where we kind of originate our thought or our decision to contract our muscle. We then send that down the brainstem, down the spinal cord, out a sensory neuron, out a motor neuron. And then from that motor neuron, we travel out to a group of muscles, we stimulate the muscles, and these muscles ultimately contract. All right, so that's a very kind of overarching, very basic approach to looking at it. And so what we'll do is we'll take that and we'll go in more detail and look at it a bit closer. Okay, so this is showing sarcolemma and myofibrils that we'll get down to in a second. And if you have access to this in your slides, you can click play and you can watch this, but I'm going to move on. So where we're going to focus, where we're going to spend most of our time is at this region known as the neuromuscular junction. All right. So the neuromuscular junction, you can figure out what this is just based on the name neuro and muscular. It's the junction between the nerve and the muscle. And this is where we're going to release our neurotransmitter, right? And so we've alluded maybe a few times to the neurotransmitter called acetylcholine. And acetylcholine is going to bind with its receptor, as we'll see in a little while. And this will stimulate the kind of passage of the action potential from the neuron over to the muscle cell, right? So this is our neuromuscular junction, the junction between the neuron and the muscle. This is the synaptic cleft in here. The way we get these neurotransmitters to release from their vesicle into the synaptic cleft is via calcium, as we'll see in a little bit. And then we ultimately have the neurotransmitter bind to its receptor here. Okay, and we'll talk about this specific kind of receptor in a second. All right. So this ultimately leads to sodium buildup, rushing into the cell. Then you have potassium leave the cell. And then ultimately you have to break down the acetylcholine because if you continually have acetylcholine at the synapse, and it's bound to its receptor, you're going to continue to have this influx of sodium and efflux of potassium. You're going to continue to have stimulation of the muscle cell, and you don't always want that, right? You need periods of relaxation. So you have an enzyme called acetylcholinesterase that breaks down acetylcholine, and in doing so, it degrades acetylcholine, it reduces its concentration in the synapse or the synaptic cleft, and this leads to reduced stimulation of the skeletal muscle fiber. And we'll talk about all this again in more detail as we move on in the different phases or the different events of our skeletal muscle contraction. All right. So our skeletal muscle contraction, and we'll stop here and then we'll pick up with part 3 in the next video. But in general, we have 3 different phases, all right? So we have events that are taking place at the neuromuscular junction, so what we just mentioned. We have events that take place at the sarcolemma, so the plasma membrane, T-tubules, and the sarcoplasmic reticulum that we've alluded to. And then thirdly, we have events that take place at the sarcomere itself, right? And so we'll talk about each of these phases in the next video, and we'll go into more detail in terms of what is happening at each phase and the specifics of how that leads to a muscle contraction.